Welcome, everyone. Thank you so much for coming today. I think it is actually the first time we're having guest speakers present here since pre-COVID. We moved into this space in November 2019. We only had 4 months. We got started with classes and then COVID hit. We had to shut it down for 2020. And then it's just taken years, I guess, to get organized again. But one of the reasons I wanted to have our own place like this is so that we could host meetings, bring in awesome speakers. And it's extra special that it's you two here today, Dr. Petzinger and Dr. Jakowec, because I went to USC for physical therapy school. And this is, a common question that I get is, why do you specialize working with people who have Parkinson's? And a big part of it is because of Dr. Petzinger's work. She was working with Dr. Beth Fisher on research, who was one of my professors in physical therapy school. And so the first thing I learned in PT school was that people with Parkinson's could get better. That exercise was really powerful. So that's all I've ever known the whole time I've been a physical therapist. And I did a residency program at USC the year after, and I got to spend some time with Dr. Petzinger in the clinic and also worked on research studies, exercise studies that were happening. And so, yeah, really this place is trying to take your research and put it into the real world. So thank you so much for being here.
Thank you. Yeah. Thank you, guys. So anyway, we're honored to be here, and also I need to also acknowledge my partner friend, Dr. Jakowec. So I always tell people, if you've got a tail, you see him. If you don't have a tail, you see me. We do a lot of rodent studies, and a lot of the original work before we even thought about human studies, and that's when we connected to. We actually were doing a lot in the lab with animal studies. So it just reminds us how important translational research really is, and we can learn a lot from animals to help validate why we do what we do, if that makes sense. I think the other thing about translation that really work is that, as much as animal work is important, right? It tells us, again, so much about mechanism. We're going to get mechanistic here. We're going to dive deep into what we think is going on. All that is important, of course, but then at the other hand, we say, okay, but why do we care? So it's meaning, how does it impact patients, right? And what does it mean in terms of patients? So the research field and translational research is very complex because you really can't do one without the other. It's nice to say that you can see changes in the animal brain, but you have to have behavior to say that it's underlying behavioral effects, right? So, and behavior, although animals are helpful, obviously we're limited. Although it's amazing what we can do with animals. So Dr. Jakowec's background is in molecular biology, and actually he was Beth Fisher's PI when Beth Fisher was a P, which was physical therapy, decided to go to the lab. And of course, I could do some stand-up comedy here on Beth Fisher, the physical therapy in the lab. But no, we had the odd mouse escape, but she did find it. But in any event, yeah. So it's really a partnership in order to understand what's going on here. All right, so I think I really like this slide. This is actually a slide that Mike put together for us, and I think the whole idea, we talk about plasticity in the brain, and Mike, actually, I'm going to let you comment about this slide.
Sure. We often think of the adult brain as something that's solidified, it's permanent, and think of it almost like a computer where you lay down certain circuits and that's it. You leave those circuits and you live with those circuits, and if you break those circuits, too bad, you're screwed. You have no more circuits to work with. And that's the downhill trend of what we think of a computer. That's not how the brain works. And in fact, the brain is extremely dynamic. And this is not my idea. This is not even Cannon's idea. It goes back several hundred years before him. There were a number of scientists who thought of things, wrote them. They didn't have the sophistication of the experiments that we could do, but we could ask very specific questions. So one thing that you've heard of homeostasis, right? All of you have heard of homeostasis. You have so much sugar in your blood, and then when you have a candy bar, it goes up, and then that's recognized by your system, and you release some insulin, and you get rid of that, and it goes down. And then sometimes it goes down below a certain level, and you release another hormone, and then it goes back up. So it's all actually oscillating. It's sort of like driving a car, fast and slow, but you're always going to go from A to B. It's the matter of how fast you're going from A to B. This is a dynamic process. This is called homeostasis. And homeostasis was a term of going back to normalcy or going back to a very steady state. And we used to think of this as just steady. It's always steady. But we know that in our car, we have a brake and we have a gas. We don't just turn the car on and go at one speed. If you did, you end up hitting things. And so the brain works in a different way. And actually, the brain is a very dynamic process. And so Cannon saw this in insulin, and he saw this in blood oxygen, he saw this in a bunch of things. And then he made some comments on the human brain. And in fact, he called this whole idea of a dynamic homeostatic process. The brain is incredibly dynamic, means that it's always changing. And we used to think, oh, it only changes in the first year of life, and by the time you're a very young adult, everything's done. And if you go out and have too much beer at the fraternity, you're going to lose a bunch of neurons, and then you're just going to suffer for the rest of your life because you've lost those neurons. Turns out that's not true at all. In fact, the human brain and brains of animals, luckily, so we can do experiments with them, study them, is a very dynamic organ, always changing. And so one thing that Cannon said back in 1929, before any of us were born, probably, is that by an apparent contradiction, the brain maintains its stability, meaning it looks the same outside, only if it is excitable and capable of modifying itself according to external stimuli and adjusting its response to the stimulation. Oh, it's always reading, it's always detecting, it's always interpreting the world around it and then responding to it. Not unlike your blood glucose is always changing. In fact, the human brain is doing the same thing. So it's a very modifiable process, and that you need to have this instability to have stability. So it's always changing. So we thought we could tap into this, that in fact, there are times when it goes maybe off kilter, and that maybe we could bring it back by the same types of mechanisms.
I think the idea here, which I really like about this, is that neuroplasticity by itself, it's really kind of this idea of change. What does that even mean? You follow me? I think Cannon kind of puts it in context back to kind of what I said. At the end of the day, who cares? Yeah, I have plasticity, but who really cares? Yeah, I have this ability to respond to injury, but who really cares? And I think the idea here is not only is it an internal thing where the brain is sensing internally what's going wrong, that may be a stroke as an example, but it's also responding to what's happening externally, right? And because it has this range, now you get the sense that it's always trying to reach this little stability. But stability happens because you do have that range. And that's why instability leads to stability, because you can constantly gauge and go back to some level of normalcy. So what is normalcy, right? We think of normalcy, if you will, as a behavior, right? The idea that I can succeed in life in kind of the bigger term, right? And also we could probably fine tune that even more to being able to move effectively in space. We tend to think about plasticity law in the context of movement in space, only because a lot of what we conceive the brain to be responsible for is truly movement in space more than anything else. It's there to help us move as hunter and gatherers, and it's also there to help us escape. That is the principal idea of what we think the brain does. Everything else, as Mike would put, is secondary, which is hard to believe, but that probably is the case. So this will also get to some of the other points we're going to make as we talk about what does exercise, what do we think exercise is doing in the context of everything we just said. If there is this idea of plasticity, if there is this idea of change from external, internal, what role does exercise play in that to help stability? Does that make sense? Okay, so we're going to talk about something very specific in Parkinson's disease. I'm really going to talk about cognition or thinking, because as you all know, there are challenges in when we have thinking problems. Obviously, we can have trouble planning our day and all these sorts of things. But I'm going to try to show you too how much thinking plays a role in mobility. And now you're not too surprised, right? Because if I've already told you that the brain is really there for mobility, right? Then it's not too surprising that even though we think of thinking as something separate, actually they are very much related and important in helping me move through space. We're going to talk also about energy, why energy is important, including the brain, and in many ways, why energy is important for helping that modifiable aspect of the brain, right? So energy is required. It's kind of the key substance that allows me to create this range to achieve normal stability. And then talk a little bit about different exercise types and how we've been able to study that in the context of energy and in the context of this bigger idea of homeostasis. This is just some background. This is just some ideas of what cognition is. The bottom line is cognition has a very specialized role, and it has a couple of different specific aspects to it that we all agree, all the language, anybody that studies thinking knows this language, right? And so we talk about different domains of thinking. I'm listing some of the key ones here. Attention, right, is a pretty important one, as you can imagine. Executive function is a huge one that we tend to talk a lot about because this guy is also affected with aging. So some of the stuff I'll be talking about is obviously very important. We see these changes in aging. We also see these changes with Parkinson's disease. The executive function plays a very important role as a CEO. That's probably the best way to describe the executive function. It's a CEO of the brain, so it's going to do all planning, strategizing. Another good way that I tend to think about it as the desktop. Okay, that's really important, right? Because I'm manipulating a lot on the desktop. That is your CEO. That's your executive system. And then we have memory, of course, which is storage. We have language, and then we have visual spatial function. Of all those ones, probably the ones that most commonly are affected in Parkinson's disease, I would say for sure, is executive function. Processing speed is part of that as well. These guys, executive function, attention, and actually, believe it or not, visual spatial. It is what makes it different from Alzheimer's, because Alzheimer's, if you guys are familiar with Alzheimer's disease, which gets a lot of attention, of course, as a thinking problem, right? It doesn't have a mobility problem, it's a thinking problem. Alzheimer's is memory. So we can see memory issues in Parkinson's for sure, but what makes Parkinson's so unique is that it really is focused more on executive and attention, and as I said, we get a bit visual spatial. Just to remind ourselves how common this is in Parkinson's disease, we can see changes in this happening upon diagnosis. So 40% of people already upon diagnosis, we're seeing some changes in there, and they're describing these changes as well. So this is just a picture that you guys see a lot. To me, it's very, I don't want to be cruel. This would be a caveman drawing right now, because this is not how we think anymore. I mean, this is your typical, certainly this is not, it's much more complicated than that. The brain, brain evolved in mobility. The whole brain is involved in mobility. It's not going to be explained by that part, right? Because you're going to say, okay, but what's happening with the rest of the brain, right? So this is where the dopamine cell changes are occurring. But the truth is, by the time we recognize Parkinson's disease, it's nowhere like that at all. I mean, there are system changes happening. By the time we diagnose this, there's system changes. Why? Because there are systems involved in mobility. And whenever, remember, we mentioned if there's an internal perturbation of anything, immediately there's going to be a change in the brain. Okay, so let's think about what Parkinson's disease is. It's a slow progressive neurodegenerative disease, meaning that there was some cell loss happening, but it's not overnight, it's been happening. Sometime, at some point in your life, there's been a change, right? And, some people believe it might happen even in childhood. No one really knows when this first cell change happens. But what's really important here to keep in mind is that brain is not going to be settled with that. Remember, we talked about homeostasis. This is happening all the time. So there's something happening immediately that's going to try to address this. So you get a better feeling that by the time we're seeing something that we can recognize, right? This has been going on for a while, and there's a lot of systems changing. And I think that's what I'm trying to really highlight here. We have two colors of circuits that I'm showing you. All I'm just trying to show you here is behavior, like movement, okay, is really a system. The one part of the brain connecting with another part of the brain. I'm highlighting two particular parts, which we'll address. In blue, you'll see the frontal cortex. Remember that picture I showed you before? That's the executive system. So there's a lot we know in terms of mobility for everybody that is using cognitive strategy. Now I'm going to use another term. Cognitive strategy is using thinking, okay? So cognitive strategy is the idea that I use my thinking part to move. That's not surprising. In fact, I'd say hello, of course, right? I'm not saying anything new here. We all do that. And it's a way, in fact, an aspect of how we learn new movements. And then we have this other part, which is highlighted in red. And the red part is the habit part, right? You know, this whole idea that we are creatures of habit. No kidding. We are. And as I always point out to people, good thing, right? Good thing, because we, you know, habit gets a lot of bad press, right? The idea that our thinking is very habitual. How many people here believe our thinking is habitual? Everyone should be raising their hands because it's all habitual. Every thought that we have, most of the thinking we have, even though we believe that we are truly thinking this through, that is not true. Not at all. We learn our responses, our cognition, our responses by emotion, our responses to anything is driven because we've been taught that. And anyone that says otherwise doesn't know how the brain works, right? Now, we may think that we thought this through, but we didn't. In fact, that's the challenge for us, to stop before a thought bubbles up in our head, to stop, right? Mindfulness is trying to get at some of that, right? Stop. Don't say anything, keep your mouth shut, listen, think, and then think about why you're feeling that way. Because a habit, understandable, it's coming from our brain. It feels like it's us, and it is, but it's a habit, right? The same thing is true with emotion, and clearly true with movement. And that's a good thing. So why? Well, because I don't know, I don't want to be learning how to ride a bike every day. Right? I certainly don't want to learn to walk every day. I mean, it's key that we are able to draw from things that we've learned. It's key for survival. I don't want to learn again that I probably shouldn't be hanging around by sharks. You know what I mean? You know, it's good to know these things, right? And so the basal ganglia in red takes care of that. Now, we agree that there's a lot of aberrancy that can happen in that system. That's all the whole drug addiction field, right? That's habit is really where the drug addiction field has to address, because when a habit forms, as I mentioned, it's good, but not always. And in the case of addiction, it's a nightmare, right? Right. And that's also true with PTSD. A lot of PTSD is around that area too. Okay. So in this case, we're talking about two systems. And in general, when you think about dopamine, which we all hear about, right? The dopamine system plays a really important role in habit. In fact, many of the diseases of addiction are targeting dopamine, like cocaine, for example, right? Cocaine hits the dopamine system, so it's facilitating certain types of habits. All I'm saying is that dopamine is really important there in habit formation. So if I have any changes in dopamine, it can start messing with that habit, and habits are learned. So let's say in this scenario that dopamine is dropping, and it drops in a very specific area of the basal ganglia because the motor, the movement, the thinking, and the mood part are mapped out very clearly in the basal ganglia. It's not one big gamosh, okay? They're very somatotopic. It just so happens that in Parkinson's, most of those dopamine changes are happening in the movement system. Not completely, but mostly. So when I lose habit in the motor system of the basal ganglia, what does that look like? That's Parkinson's, right? That's exactly what that is. It's slow. So it's not like you actually lose the habit, which is what you would think, right? You don't lose it, but you get slower, right? And so that's fundamentally what Parkinson's is. It is the loss of a habit that makes me move slowly, right? But remember, I have homeostasis, right? I have a system, right? And the system is trying to reach stability. So I can draw from another area, and that other area is the frontal system, right? The executor or cognitive strategies. And we talk a lot about that because it's used a lot in exercise and rehab, which is really trying to help that aspect of the systems, drawing on cognitive strategies. Does that make sense? And remember, those cognitive strategies will involve executive, they're going to involve attention, all those things. I have all that access to myself to improve, right, my mobility through space, right? So remember that, right? I think one other system that we're not going to have a chance to talk about this today, but there's a lot of cool work being done in it. And I do show a little bit of it, and that's the cerebellum, this guy back here. Cerebellum is involved in balance coordination Anybody that's done a lot of ballet, whatever, you've got that probably tripled in size. I mean, it's very important coordination. It's what's really cool about it is that there's clearly now a lot of data to suggest that that's also getting recruited very heavily in a systems way when my basal ganglia is not working very well. Isn't that interesting? And it can do that in a lot of different ways. Keep in mind that the cerebellum isn't something that you're going to be as aware about. You're not going to go, oh, let's activate my cerebellum, right? That's not going to happen, right? But it does it automatically. It's called implicit. It's an implicit aspect of it. So it's going to try to help. Again, lots of work going on to show how much it helps. But clearly, animal data showing that if we put an animal on an exercise treadmill, that thing is lighting up like a light bulb trying to help. So that's really cool because, again, these are, as we understand this whole idea of homeostasis and who the players are in it, it gives us more breadth. It gives us chance and opportunities, right, to see what else I can do to maximize, you understand, this range, right? Because, I think the idea that we're promoting, we're still trying to help the habit part by the dopamine replacement, clearly, but that I have other players that are also coming online. Does that make sense? And we believe that really probably at the end of the day, probably one of the reasons it takes so long, it's not the only reason, but one of the reasons why it takes so long before we recognize what the is could very well be these systems, you see, which I find very interesting and very cool from a kind of a logic experiment where I would say, okay, let's see now. If I am looking for therapeutics, right, here I have two possibilities. Let's see. One, I can recreate the brain, recreate it, go back to 40 years ago when your brain was 40 years younger, or I could go over here to the side of compensation, understand what these systems would try to do. Yeah, I don't have all my dopamine, but I'm still successful, right? And work on that. You follow me? I don't know about you, but I'm kind of a big believer of, if I've got somebody that might be working, why not use it as opposed to trying to get back to a 40-year younger brain. I think that's harder, right? And I think the PD field has a lot of soul searching to do in terms of recognizing what's even doable here, right? Recreating a whole brain or maximizing, right? I like to say that a lot of the work that we're doing is trying to understand how to maximize what? The homeostatic range, you see. Now you understand. I want to maximize it because if I have and can maintain a homeostatic range, if I can get those sources stable where I can draw from them, I have some other way to solve the problem. Do you understand what I'm saying, right? I will get to talk in a second. But I think the one thing, though, which is such an important idea, this is called the elephant in the room. So the elephant in the room, guys, is aging, you see. That's the elephant in the room here. Because the brain ages. That's a whole separate thing here, right? What do we do about the aging problem? And a lot of what we talk about here isn't just simply about PD, it's about the aging problem. Oh, right, I've got to prevent these cells from aging, right? Good luck on that. There's a lot of people that are working on nothing but aging. And now you know why, right? It's something that all of us are facing. And oh, right, it may be the key factor here in terms of what homeostasis is all about, right? What my range is, right? Why can't I maintain it? May have everything to do with aging, period, right? And who's talking about aging, right? Yeah, that's an old topic that comes up time and time again, but now you begin to see that range of studies going on. You got it? Because you've got people over here that are trying to rebuild the brain, telling you that all I need to get is dopamine cells back. You have people over here looking at aging. You have people over here looking at compensation. So there isn't one way to look at this problem. You see what I'm saying? It gets complex very fast. All right. So what is resilience and how does this fit into this? Again, I want to put this in the context of homeostasis. We're keeping the term homeostasis front and center. So whatever I say now has to fit that model. So resilience in general is thought to be a loss or diseases, if you would, are thought to be loss of connections. And that's just a very simple concept. I'm now losing my ability to have the systems working together because they're physically not connecting anymore. Now we can get a little fancier. We can say they're functionally not connecting. So physically or functionally, they're just not working together anymore, right? For various reasons, disease, aging, etc. So the concept behind resilience is this: I am gathering more connections, so I'm physically adding more connections right there, or I'm making whatever connections I have, stronger. Now, again, if I had to put that in the context of homeostasis, it would be the idea, again, of range, right? I have other systems that I can recruit in. Again, that huge flexibility. I want resources. I want access to resources, and I want access to resources in areas of the brain that I think are key, right? So this whole idea of connections is really about that. It's about physically having access, creating more connections, or making those connections stronger, so I can draw from them to make mobility and behavior better. We have examples of this happening, and I always like to use clinical examples. Mike, can you talk about the nun study?
Yeah, just like, shoes got a problem in the lab.
Oh, got it.
Let
Okay,
me fix this.
go ahead. All right. So in the nun study, what this was is, and I'm going to back up a second before I talk about the nun study to remind ourselves one other point. In many neurodegenerative diseases, such as Parkinson's disease, there is thoughts about what's causing it, the etiology. We would say genetics and environment for sure. But there's also something that we can see visually that's happening in the brain. We can certainly see dopamine changes. We can see that there's less dopamine. We can also see an accumulation of a protein that's appearing there. It has a normal function. It's just, we can now see it. It's like aggregating out. It's like precipitating out of the cell, so that it's suddenly what wasn't visible is now visible. And that's called alpha-synuclein. And alpha-synuclein is seen. That's the name of the protein. And again, it has a normal function, so it's not like it never had any role there. Anyway, we're seeing it in areas of the brain, such as the substantia nigra, where the dopamine cells have degenerated. And we see it not just there, but we also see it up into the thinking parts of the brain as well. And we do know that we see some accumulation of that over time. So you have it accumulating in one part of the brain, and as time goes on, it accumulates across the brain. The role that this protein plays in Parkinson's is pretty clear that it does. I mean, there's a lot of really nice data to show that somehow it is contributing to some of this loss of connections and loss of cells. It's probably not the whole story, definitely not the whole story, but it is a player in the story. And so there's always been the interest in thinking about what would happen if I could remove the protein, and would that be a good thing or not a good thing? Alzheimer's disease has the same story, but their protein, they have two. It's called amyloid and tau. Same idea, exactly the same idea, except the accumulation is a different part of the brain. And this is just to point out the idea of what resilience could mean. Because in this nun study, what they did is they followed these nuns for a number of years, and they were able to track their level of thinking, in this case, because it's Alzheimer's, by their writing and their grammar and these sorts of things. And they were able to determine how robust their thinking was based on that. And then at the end of their lives, they gave up their brains. And in that study, what they found was that there were a number of examples of individuals whose brains were full of amyloid and tau. And yet their thinking did not reflect that at all, meaning that their thinking was still quite good. I think it just reminds us, again, in terms of what resilience can mean. It wasn't that they didn't have the pathology, they did. But the idea that these connections that they had were very efficient, or that they had more. Does that make sense? And that also gives you a better idea of what resilience could mean. It's not just the idea of more connections, but it's also the connections in the context of even having disease. So resilience isn't an independent phenomenon. It's happening in the context of what? Injury. Does that make sense? So resilience is what's providing the robustness of the system, even though there's something ongoing. And that's a good way to think about what Parkinson's is. The truth is, though, I can blow that up even more. I could say that's what we're all going through. Parkinson's is one example of that. Alzheimer's is another example of it. But there is many things that our brain gets, that we are exposed to. I mean, nobody lives in a bubble, right? Everyone has, if you live in California, I'm from Northern California, I'm pretty sure I've met my quota on pesticide exposure, probably over that, right? Most of us have. So that's the big point here. I mean, I think the idea of resilience is that whatever the pathology is, not just now, but for the rest of my life, right? Because this is not going to stop. Whatever these exposures are, it's not going to stop unless, you move on. I don't even think Mars anymore, right? Right? Anyway, that's gone. The point is that is what resilience is. So what I like about that term, it's that it's a breathing term. It's a real term, meaning that it's existing in us all the time. And in a way, it's the best term to talk about what homeostasis is. So we're given a term of homeostasis, which has a very science idea, doesn't it? But now I'm going to make it a behavioral idea. And I'm going to say, this is what resilience is at the end of the day for all of us. It just, in this context, we're going to talk about it in Parkinson's. Sorry.
Okay.
I'm not going to show this because I think you guys have, yeah. And this is just to remind us of some original work. This is some of the oldest work we ever did. We were the first group at SC to look at animals that were Parkinsonian. And the question that we were asking was whether animals that were Parkinsonian could receive and get better. So this is the whole idea of using an external stimuli to see if we could build results, right? We knew that dopamine cells were lost in this case. We knew that there was dopamine depletion. So the question is challenging. Remember that statement by Cannon? Challenging it, the system. Can I do something to boost homeostasis? That's what we're doing here. And so that's some of the first animal studies that we did. This is a really big study. And this is just to show behaviorally what we were seeing. And right now it kind of seems so intuitive, but at the time, everything we're talking about this would have been, Martian to anybody in the science field, certainly in the Parkinson's field. Parkinson's was thought to be like over here, right? And then there's something over here. And I'm hoping what I'm trying to do is tell you there's no here and there. It's the same thing. You understand? All the language that we're talking about isn't necessarily even Parkinson's specific. It's injury. You follow me? Does that make sense now? And so the rules of the game, I always tell people, no one gets a Martian brain here. It's one brain. We got to make that model so that I can talk about that model in any system that I want. And it still makes sense. You see what I'm saying? Right. So when we were first doing these studies, that's what we had to do. We had to say, guys, really, you're embarrassing us. Parkinson's is the same thing. You see, it's a systems problem like anything else. So we're going to think about it that way as well. So the original work that we showed, we're basically starting with dopamine systems just to remind ourselves, yes, we could see that we could change even the homeostatic characteristics of dopamine. We could actually make dopamine receptors become higher expressed. And the way we did that was this is an animal brain here. This is the rodent brain. This is cutting off, so this is what we call a coronal section. So I'm cutting off the nose. I'm looking deep into the basal ganglia. In red is dopamine receptors. Dopamine receptors are key for habit. Dopamine works on dopamine receptors to make habit function, right? So think of a car, gas is the dopamine, the engine is the receptors. Gas hits the receptors gas hits the engine like dopamine hits the receptors, and now the car moves, okay? Think of it the same way. So that's what we were basically, this is normal, lots of dopamine receptors. Here's a Parkinsonian animal, and this is with exercise. Nothing else, just simply exercise with the key being, hello, Cannon's rule is happening. It can totally do this. If we drive it, if we provide some external stimuli. Now remember, this is also key. I think this is so critical that it gets missed. It's all about what? Challenge. You see, why give that homeostatic range if you're sitting in front of a TV? I'm not going to give it to you. Why would you need it? The brain doesn't know what it needs. It's not a magician here. It exists because it's being what? Used. You create that homeostatic range. You create it because you're asking the brain to do what? Do more. You understand? So that homeostatic range only happens, yes, okay, something internal, but the external challenge telling that brain, I need more here, guys. I need more range is really what's interesting, isn't it? Fascinating, really fascinating. Because the rules of the game, how much range can I get here? You see what I'm saying? That's ginormous. And not to keep getting back on a little soapbox here, but whatever. Get used to it. The point is this. I mean, I would argue, again, if you look at studies for Parkinson's, what are they doing? Even, I'm telling you, even these gene therapy stem cells, there is no component of these studies where there's any sort of lifestyle or exercise component at all. And you're thinking, how is this supposed to happen? You follow me? Because it's not about just getting nutrients or anything like that. The brain needs to know what you're trying to do. It doesn't guess this. It doesn't go through gene therapy. You sit there and it goes, right, let's see. I want to ride a bike, I want to swim. You know what I mean? Why would it do that? Cannon's law exists. It only knows that because you're telling it, I need this range. You see what I'm saying? Isn't that fascinating? Right. All of these are just basic ideas of how the brain works, but become critical when you think about designs and what it is that you're trying to do.
Do you think that's kind of why a lot of studies have failed, is because they were missing like the stimulus that could have maybe made that treatment work?
Yeah. Yes, that's right. I mean, believe it or not, there was a trophic factor study, which we'll talk about trophic factors in just a second. And this is trophic factors help a little bit. What they do, they're one component of synaptogenesis of connections. They were literally just putting the trophic factor gene in the basal ganglia. That was it. They just put the gene in there, make trophic factor. That was it. And you're thinking, okay, I mean, isn't this like the Matrix film or something like that, right? Where, okay, I just put this in and suddenly I know this. I'm going, how? How is that supposed to happen, right? We need Cannon's law like front and center, guys. It's not going to happen if the brain doesn't know what to do. And I think that is true. And again, I think I really like the idea, again, of homeostasis, because it is really what we're trying to do, isn't it? That range, the range that we're trying to create. And yes, we can, say, can we always use help? Of course, we can always use help. And maybe that is where the therapeutics really are. Help me get that range. I need that range. I'm trying to create that range because I'm expanding my experience, but I can get some support here. That makes more intuitive sense, doesn't it? Than just saying, here, good luck on that. You know what I mean? That makes no sense. So we saw the same with humans. We saw, this is a study that actually, I think Claire, you might have been involved with this as well, but you and I think maybe certainly also Janine Yip. All I'm showing you here is it's a subtraction scan where we took images of the dopamine system, the receptors before and after 8 weeks of exercise. And red means an increase. So the red means we were seeing increase in dopamine receptors. So we basically repeated exactly what we saw in animals, we saw in humans, right? So here's the translational idea here. We start with an animal model, we figure out what are we looking for, do we see it, and then we replicate it. You follow me? So it's a one-to-one experiment. I'm trying to show this. This has been now shown by other groups. Well, actually, really one of those groups, like John Stossel in BC has done a little bit of work on this to replicate what we have reported. So how do those connections? And connections are very happening in a very very small. I mean, you can't image this. So people often ask me about any type of imaging we can do. You can't image this in humans. The MRI is not showing you this. There's no divide. There's nothing out there right now in humans that would allow us to look at connections like this. So we have to do these in animals. We have indirect measurements of it, but we don't have anything direct. So we can't in a live human, at least not yet, be able to measure connections. So this is.
Yeah, what are you talking about being able to measure? Like what kind of connections?
Oh, these are between neurons, cells. So remember when we go back to the idea of resilience, we're talking about getting more connections, more efficient connections. These are between the main information gathering and coding part of the brain called the neurons. So neurons are what's connecting things together and encoding information. Does that make sense? And depending on where they are, they play a different role. So as I said, the basal ganglia, they're what's encoding the memories of habits, right? And in those different domains of the brain, they're encoding that type of information, whether it's attention or those sorts of things. Okay, so we did some studies in animals again. So this is just the kind of layout of the lab. Here's the basal ganglia here. what you're seeing in black are these are neurons throughout the brain. We fill them with black dye. And what's marked out here in the hatch is the basic ganglia. If we blow that up, part of the basic ganglia, blow that up, here's a neuron right here. And this is just to show you, it has a sort of arborization. So the connections aren't really made on their cell body. They're actually made on all these little outstretches, like a tree. It's like a tree. So it has a lot of these branches, and all the connections are happening on the branches, not on its little cell body, probably because there won't be as much room. You can't get as many little connections on a little ball, but you can if you have a lot of branches. And so here, this is one of our graduate students, just to show you some examples, filled and then colorized it so we could see the differences. This is a normal looking neuron. We don't see the cell body, we just see the arborizations of it, where the connections are going to occur on these little branches. So that's a healthy neuron in the basic ganglia. This is what happens with dopamine depletion. So we see that we get some loss of that arborization connections, some physical loss. And then this is with exercise, more arborization. And we decided to also take a look at it, not just from arborization, but counting, because each one of these guys has tons. So if you see this little line here, see that's what this line here in purple, for example, that would be sailing. That would be that branch. And then see where all the connections are. It's not the connection, but it's where the connection will happen. It's the landing dock of where a connection will happen. So here's sailing. Same exercise, we actually see not just the number, but maybe some of the shapes. It's kind of elongated. Here's MPTP, and with MPTP, you can see a bit that it's a little bit more denuded. So we're going from here to here. Here's a red branch. So here's the branch again. We see also that the branches that are there, they're not as robust, the connections, right? And here we have exercise. We call that synaptogenesis. That's what we call that. So it was basically trying to show that we can build resilience by driving the system, right? So the idea back to Kennan's law, challenging the system is what gives us that range, and that's what we began to see. Mike, I'll let you demand politics.
Oh, can't have
No.
But challenge the system.
So what we're transitioning to is trying to understand the energy of the brain. One thing that's interesting is that in many diseases, Alzheimer's, Parkinson's, there seems to be a reduction in the energetic state of the brain. If you go in and you use imaging techniques, even very crude imaging techniques in humans, you see a reduction in some of the metabolism. Glucose, for example, or ATP, these markers of energetic demand are being reduced. And another thing that we thought was very interesting was that there's a number of diseases of the brain, especially ALS. ALS is Lou Gehrig's disease, amyotrophic lateral sclerosis. It's what Stephen Hawking had. And we always thought of it, it's a motor neuron disease, therefore it must be a disease of the neurons. And people have thought that way for about 200 years in terms of it's a neurological disorder, therefore it must be neurons. And continuing to hit our head against the wall and not finding anything that's relevant, they in fact stumbled upon the fact that maybe it's like one of the cells that's supporting the neuron that's sick. Same thing happens in MS. MS, we thought of, it's a disease of the neuron, but yet the cause is in the oligodendrocytes, the cells that wrap the neurons and put that fatty tissue around it, and they get damaged and then open up the door for neuronal damage. So the gatekeepers have been compromised. Same thing happens in ALS. It's actually the astrocytes, another group of non-neuronal cells, of what you have as many non-neurons, probably have triple the number of non-neurons in your head or in your brain as you have neurons. People used to think that they were just there for the glue and the scaffolding, they're just holding up the building, holding up the organization of the network of neurons. Turns out not to be true at all. They're in fact extremely important for supporting the life of a neuron and communicating with neurons. The energetic state and things of that nature. So we began to ask very specific questions. One thing that also interested us is at the same time, understanding the link between the body and the brain, the chemical link. What is it? There's no spiritual link or some, ghostly thing. It's not, this isn't Descartes thinking of the spirit in a machine crap. This is actually the whole idea of what is it that tells your brain that your body is moving. They think, okay, that could be important because maybe you need a mechanism to report to the brain and then maybe to select and strengthen those circuits that you have been using to do that motor behavior. Kind of makes sense. We do that in early life. We try to think of your first year of life. Think of it when you're 9 months of age. Just think back. And remember that in fact you were crawling around, and then you got up and you did some furniture surfing and you started moving around. And then you started getting a lot of reward because, yay, I can now reach the cookies. And you're getting a lot of feedback. Mom and dad were clapping and applauding, like, wow, that is so good. You're just, oh, wow, you're walking. This is amazing, incredible. And then you go, yay, yay. And the brain sees and feels this yay, yay, yay. And says, well, what is it that you just did? And can I keep those circuits that you just did? And can I strengthen those circuits? Because that behavior that you just did is actually something that I may want to keep because I'm getting all this reward. I'm getting cookies and I'm getting happiness and I'm able to go around the house. Wonderful. And you'd like to do that. When we were thinking of what is it that does that? Does anybody exercise? Exercise? And do you ever get muscle cramps?
Yeah.
Why? That's I like that. You don't know why. Why do you get muscle cramps? Think about anybody do. Oh, lactate is it? Yes. And so what happened? The coach says, walk it off. Get that lactic acid out of your muscles because that's evil, right? It's horrible stuff. Want to get rid of it? Anybody drink wine?
Yes.
Okay, a couple. Red wine's good for you. Of course, you know that. Louis Pasteur back in the 1840s went to the Loire Valley, and people there were having trouble making wine, and they were making vinegar. And he's like, well, we don't want vinegar, we want wine. And so he began to study this metabolic process. When you make wine, you're actually converting sugars into something else, right? And the sugar is converted into something like, well, ethanol. Okay, that's good for making beer and wine. It can also be used to manufacture. Anybody make sauerkraut? Pickles? No old Polish people around here.
Eat it.
Come on, guys. Okay, Kapustka. Those of you who make fermentation, when you ferment, you actually also make lactic acid, and you make pickles and all that kinds of stuff. So Louis Pasteur laid down this whole foundation that the production of lactic acid in wine is bad, and it's a process that only happens when you don't have oxygen. And when you don't have oxygen, you start doing other things, and you break down glucose into an intermediate, and then you end up with either ethanol or lactic acid, and that's it, you stop. And then, when you exercise, such as you're doing a 400-meter sprint, you make a lot of lactic acid, and a lactic acid, because you don't have oxygen in your muscles, right? Because you're using all that oxygen to immediately, and then you run out of it, and you convert over to this anaerobic glycolysis, a type of breakdown of sugars without oxygen, and then you accumulate lactic acid, and you want to get rid of it, and it's bad, right? Complete BS. Flip that around. It actually turns out that it's something that's very good for you. We start thinking about a mechanism that allows the body and the brain to communicate. And it's actually through lactic acid. And actually, it's not lactic acid, it's actually lactate. What that means is that it doesn't cause a pH change or an acid change. It's actually a molecule that we make. So this is starting to make some sense. Now that your muscles are moving and you're doing something, and whether that's running, walking, or doing very specific exercises, and you're doing something, and then you're generating a lot of lactate because your muscles are breaking down glucose, sugars, into, and you go back, think of your biochemistry back in high school. It's been a couple of years. but breaking down glucose into a whole bunch of intermediates, and then pyruvate, and then into lactate. So we make this lactate, and then we spill that lactate out of our muscles, and it goes into our bloodstream. Where does it go? At actually 25% of all your blood goes to your brain. So now the brain sees this incredible burst of lactate, and sees all of this.
Next, that's the.
Oh, is that the next slide? Okay.
But just to point out again what Dr. Jakowec was trying to bring up. We talk about energy, right? Mike is bringing up two very particular points. One is we're trying to say we need energy. Where can we get it? He's already giving you a hint. It's going to be coming from the periphery, but it's not glucose. We think of glucose, right? Everybody knows glucose as probably the big energy. But what he's trying to tell you is it may not be that. Actually, it's lactate. It's really not an acid because it's not acidic. It's a compound that we all make when we're active and excited. That's called lactate. I think the other point that Dr. Jakowec is trying to make, which I think is also very clever, is this idea. Remember, we're doing this challenge system. And the challenge is a physical challenge, and yet how does the brain understand that? There's a lot of different ways that one could understand that's being translated to the brain, but the argument now is that it could actually be even the chemical itself. And we're going to hint at what other things lactate can do besides being a fuel. And that's why this is so interesting, the idea of lactate.
Yeah, so in fact, the amount of lactate in your bloodstream that's in your blood can go up like 300-fold. And it can be far in excess of what the glucose is. That's like, wow, that's kind of weird. The regular sugar that we think we use in our brain is actually not the main fuel of the brain during exercise. And in fact, it's something else, and it is lactate. And this lactate is something that is actually fed to the, I don't know which button. Oh, there it is actually fed to the neurons. The neurons are chugging away. They need to be fed. And it turns out that they're fed because of another cell that is well positioned. And this is called the astrocyte. And the astrocyte is really quite remarkable in that it looks like a giant spider, sort of like this giant monster here. And here we have the neuron, of which you have maybe 86 billion neurons in your brain. And then we have these astrocytes. We probably have 150 billion astrocytes. And one single astrocyte can in fact monitor almost 2 million connections within the brain. And you're like, whoa, that's pretty impressive. It can also dictate and is responsible for the blood-brain barrier. In fact, it's kind of interesting because here it is making connections to the neurons. It's also making connections to the bloodstream and is a bridge between the bloodstream and the neuron.
Right, so
And
this
it's
is an
feeding the
important
neuron.
point. Pardon me. Fuel isn't going directly into the neurons, you see. It has to go through astrocytes. Isn't that interesting? So it's not neurons that are, when you see blood vessels going through the brain, they're not actually going directly to neurons at all. It's astrocytes that they're going to. So astrocytes, another way that I think about it is astrocytes are like gas station. They're the gas stations, actually. They're the ones providing gas to everything. And they're well situated to do that because they are so ginormous and they can monitor. Remember all those connections we're looking at. These guys are paying attention to them. So that's why astrocytes are so important.
Yeah, so these astrocytes, we used to think, a lot of people still say it. And in fact, even I read a paper yesterday by some other scientists around the country, and they still said it, which was the fact that when you exercise, you deliver more blood to your brain, right? And therefore you bring a lot of nutrients into the brain, right? All over the brain. And you get rid of things you don't want, waste from all over your brain. So you're basically making a healthy brain. And I'm thinking, and our evidence shows this is not true at all. Anybody garden? You garden? What do you put on your garden? You can say the word bullshit. Go ahead. You're amongst friends. Oh, you feed it. Okay. Now, when you feed your garden, and let's say your garden's the size here, do you throw bullshit everywhere? Like all over everything? Like all over your peas and your carrots and your tomato plants here and on the roof of your shed and on your friends, just go everywhere?
No, I discriminate.
Oh, so you're very specific in where you put your bullshit. Okay, excellent. That makes sense to me as a gardener because now you can focus the manure on your tomatoes because you don't want the weeds over there you don't care about. So what's happening here is that you are actually feeding a very specific part of of your garden to get what you want out of it.
Yeah.
When we exercise, we actually feed a very specific part of our brain. It doesn't open up the whole brain in order to get this giant flood of blood. That's called orthostatic hypotension, and you faint. What's actually happened, this is something that we discovered a few years ago. It's really interesting. I'm telling you that there's 86 billion neurons, 100 billion astrocytes touching every single blood vessel in your brain is everywhere. However, when you exercise and when you think and use very specific circuits or very specific combinations of neurons, the detection, the relationship between those activated neurons and the lactate that's coming in is not occurring everywhere, it's occurring at that particular site. In fact, blood flow changes. Blood flow, all those little vessels actually open up where they're needed because you're exercising. And they can bring in the nutrients, glucose, especially lactate, to get rid of things, specifically in that one particular region. These astrocytes, and we have data that shows it, is that these astrocytes in neurons that are being activated actually respond very differently than a neuron a little further away that's not involved in monitoring these circuits. And in fact, they begin to change. What they'll do is they'll elevate their capacity to transport lactate from the bloodstream into the neuron. And so they can specifically feed these particular neurons. Wow, that makes sense. That's the manure that's feeding your tomato plants and not feeding the weeds and all the other plants in your garden.
So also just to point out, as Mike is trying to say, this effect is very specific and it's circuit specific. If I'm using some part of my brain, it's only going to do that. And the astrocytes, being the detectors that they are, sense that. And that keeps it from a generalized effect. And as Mike pointed out, it's not unusual to see in the literature this whole idea that we're activating the whole brain. Mike is just trying to tell you it's much more finesse than that, for all the reasons that you can, for all the obvious reasons. Astrocytes are situated in a way that they can do that. They're really fine-tuned because they are detectors. At the same time, they're detecting where the need is based on how these neurons are being activated. I think that's also really an important point because in general, we tend to think of the brain as sort of this recipient, like a sponge of some sort, right? It just bring it in and I'm going to use it, whatever. I pointed out that will never be the case. One, for example, the brain doesn't know what to do with it, right? Necessarily. It needs to be part of this equation. It needs to say, this is what I need because I'm challenging the brain in this way. It's that same challenge that's beginning to direct and tell, this really cross communication, which is really cool, between what the brain is doing and what the body is doing. And the brain gets specific about it. Like, no, it's this part, guys, over here. I need blood flow and I need fuel. And that's really, really cool because it begins to help us understand how specific this is and how and what's directing it. Does that make sense? We're just trying to kind of teach you guys and help you understand better really what we're understanding today in terms of what the data is showing. And again, the fact that the astrocytes are so, they're the obvious character here. That's what we're trying to say. They are the obvious character. They're well situated. Not only do they manage lactate that's coming in, but they also manage glucose that is coming in that can be converted to lactate, right? But the lactate story is a relatively new story because you won't see that very much right now, but it's coming. That lactate, which we thought was bad, is not bad. In fact, it's a key molecule.
And the other key molecule, so just like your manure is bringing nutrients to your tomato plants, it is also bringing other things to enhance that growth. And exactly what astrocytes do. They're bringing the fuel from the bloodstream to the neuron, but they're also bringing the neurotrophic factors. You've all heard of BDNF, GDNF, CNTF, this magic dust that they sprinkle into the brains, and all this amazing things happen, and they're trying to do therapeutics for it. In fact, what happens is that this relationship between the astrocyte and the neuron and bridging the bloodstream, when it is involved and activated, we're trying to figure out all the molecular rules right now of why it's being activated and how it's being activated so that maybe we can get a therapeutic that can make it even stronger, is that in fact, when that relationship happens, it then also makes compounds that strengthen that relationship and feed the relationship. So in fact, you will give nutrients, not just immediate nutrients like you need right now, like lactate and glucose, sugars to burn right away, but it also will give you nutrients, neurotrophic factors that will change the structure. And you heard about synaptogenesis just a few seconds ago. And in fact, it will enhance synaptogenesis and strengthen those connections such that they will be long-term or maybe even permanent. So it's almost like identifying a relationship that, yes, it's like going on a date. Okay, you go for it, like you do speed dating, you go your first dinner, and then you have your first dinner, you get fed, and then, okay, this is our relationship. So I'd like to make it permanent. We're gonna take it to a different level. Astrocytes are like dating. Okay, they first feed you and they say, this is a nice dinner. Oh, do you want to continue this? Sure, let's do it.
So-
And they create this relationship. And this is what
We're
astrocytes
going to show the data.
Yeah, okay.
Okay, so I know that was a lot to take in, but I think, again, obviously we all agree astrocytes are important. And just the capacity of astrocytes and many of the unique things it does is probably going to be a pretty key player here. And again, the idea that we've been focused on neurons, which is, neurons are important, they're what we're looking for. At the end of the day, it's about resilience. Those are neurons. But the question is, what is giving us this range, you see? And that astrocytes have a lot of ability to do that. So we looked at just one study that we did. This is a work done with one of the Petzinger one. They were just looking to see what happens with astrocytes over time, even with exercise. And I think one of the things we saw was that they got bigger. They certainly got larger, and these branches got even stronger yet. So we thought that was kind of cool, that astrocytes, not just synapses were forming, but that these astrocytes were getting bigger and thicker. And we picked two areas of the brain, the PFC, which is the executive system, and the striatum, which is the habit system. So we saw it in both these areas where astrocytes were clearly getting bigger with exercise. Kind of nice, because that's what we're hoping, is trying to see if astrocytes are changing with exercise, if we think that they're a key player. I think the other thing that we were interested in was these two ideas. I think one, and we'll just hang on there for me for a second. One is this idea that we're getting lactate with exercise from the periphery here, right? So this is coming in from the muscles, lactate's released, going into the blood, crosses the blood barrier into astrocytes, and astrocytes are feeding neurons for sure. I think the other thing that we found, we're trying to suggest is that with exercise, we're also getting increased neural activity, meaning certain circuits are getting activated. Here's a neuron, for example. It's getting activated. It needs more energy. The neuron is firing. It needs more energy. It actually starts also changing, getting ready for more astrocyte activity. So all we're trying to say here is that the two are happening together. Astrocytes get activated, and neurons, because we're demanding them to get involved, get more activated. This is just the idea of what we expect to see with lactate. This has been already reported many times. We can see this elevation of lactate quickly with exercise. And the other point that Mike was bringing up here is sort of the timeline, and this also very much fits the diet field when we're interested in looking at what role diet can play. But this is just the first thing that gets broken down when you're exercising, where the sources of energy are coming from. Glucose is the first thing. So glucose is coming in first, right? Whatever glucose stores you have, they're readily accessible. They make it to the brain, and we're saying that glucose, even when it hits astrocytes, are probably getting converted to lactate and uses lactate. Lactate is probably the principal fuel in the brain. So glucose, even if it's glucose coming in, into astrocytes probably get converted to lactate, and lactate is helping the neurons. Does that make sense?
Yeah, and while we're sitting here, our brain is mostly using glucose. And that's sort of like the in neutral rep, just kind of like stable position right now, because you're not really doing anything in terms of motor or exercise. You're just maintaining. We use glucose.
The next source of fuel that we'd be using for the brain is lactate, though. That's second. And that's going to be particularly true when we start exercising. And we don't have, if we're exercising for longer, so here's time, then we start recruiting fats, and then eventually start breaking down protein. So that's sort of the order of what you would expect. So for low intensity, as Mike says, the first thing, because it's the easiest accessible to sitting, it's going to be principally glucose. Then sedentary behavior is principally glucose. Again, down in the astrocytes, it probably gets converted to lactate. And the next thing, the next type of fuel we'll be using then is lactate. And what's cool about lactate is it's very different from glucose. So glucose is predominantly just for energy. Look at all the things that lactate does. It's like, boom, wait a minute here, okay? It's doing a lot of other things suddenly. It's not just giving fuel, it's creating an environment to make it last longer. And that's what Mike was trying to say. It has this immediate effect, but it also starts preparing for a long-term effect, saying, wait a minute, this is big, and I need to start thinking about how I'm going to start remodeling this relationship in these neurons. And then the idea, so we're principally interested in lactate now. This is our son, so not him. Anyway, he's a USC runner. But anyway, we're just trying to show that, glucose and lactate basically can come from the muscle, a little bit from the liver as well. And ultimately, what we're trying to do is support this circuit. And it's being done through exercise, again, just to remind ourselves through exercise, muscle, lactate as a fuel. Lactate is energy fuel. We also talked about trophic factors. Lactate can help these synapses and it can grow. We also know that lactate can actually increase mitochondria, and mitochondria are the energy producing part of the cell. So you're talking about some pretty big things. Again, if we're trying to create range here and try to figure out what molecule would be pretty good one to look at, lactate would climb pretty darn fast because it can do a lot of range and hit it at multiple levels. From a fuel point of view, from a connection point of view, and even mitochondria, which is a powerhouse for energy. So this is some work that was done by another grad student in the lab, and these are astrocytes that he actually isolated from mouse brains. Some mice were run. So this is we didn't do this in humans, but we're just trying to show that human running is this rodent running. So lactate is building up in the muscles of this mouse. We also did some parallel studies actually injecting lactate in sedentary animals to see if it's lactate. I can get lactate because I'm either running or I can inject lactate. It should give us the same results in theory. So we move out the astrocytes, we take them out from the mouse brain, we collect them and we see what they're making. And we were able to show these are a number of different trophic factors, and absolutely these astrocytes make trophic factors. So the theory of what we think should be happening, we're proving this in the animal. It's absolutely true. Lactate is most definitely increasing trophic factor levels in these astrocytes. So it's when we talk about exercise, you often read that exercise is increasing trophic factor levels in the brain, and we think it's good because it's a fertilizer, kind of that concept. We're now telling you where this is happening. And not only where this is happening in exercise, but how it's happening. And it seems to be lactate. That's the major driver here. And I think the other point is now you're also beginning to understand how the two can be connected, right? How does trophic factor happen? Well, it is this idea that I'm becoming more physically active. I'm now recruiting more lactate than glucose because I'm exercising. It gets to the brain. It's getting these astrocytes bigger. It's causing a signal to these astrocytes to start making trophic factors. That trophic factors now being delivered to neurons. Does that make sense? You're getting a much more better understanding. And I think to Mike's point, remember, because the astrocytes are sensors, they're not going to let this happen just willy-nilly. It's going to be needed. They're going to sense where this activity, where this energy needs to be delivered based on what I'm using, what part of the brain I'm using. Does that make sense, guys? Yeah?
The brain itself, I mean, if you looked in the brain and said, okay, while you're exercising, I see these neurons being activated. Okay. Now, if you went to that neuron and said, neuron, what are you doing? It probably doesn't know what it's doing. All it knows is that it's activated and somewhere downstream, it's having an effect somewhere. Okay, cool. Then you go to another neuron and it's being activated and you say, yeah, neuron, do you know what you're doing? And say, no, I really don't know what I'm doing. But then you look at what it's doing, it's actually involved in thinking. Oh, that's kind of cool. So we got some neurons that are doing thinking and some neurons that are doing moving, and maybe when we feed the fertilizer into the brain, we can actually strengthen those neurons that are involved in moving and thinking. Oh my gosh, could this be something interesting? Like maybe it's not just moving, but when we think and move, and I think this is the whole basis of why you're here, is the fact that you are engaged in your exercise. You're going to get
Yeah.
Okay, I'll stop there. Get ready for it's gonna make sense.
This
Yeah.
is our evening meal discussion.
If I give away the punchline, she hits me on the way out. I'll shut up.
Yeah, but I mean, so we're back to mitochondria. We said, mitochondria powerhouse of the cell. It makes a lot of energy. Lactate makes energy independently of the mitochondria, but also does it through the mitochondria. We are very lucky. We work with a very talented researcher who can map. What he does is he can image certain markers that we want imaged. In this case, he's mapping mitochondrial function. And this is an animal exercising. And all we're trying to show here is that this is the difference between an animal that's non-exercising and an animal that's exercising. We're showing parts of the brain that are lighting up in red, in yellow. These are mitochondrial activators. And I think one of the things you can see is that it's clearly not the whole brain. I mean, there's certain areas of the brain that seem to be lighting up. You're going to see here in the center part of the brain, starting with the basal ganglia. So you can see lots of parts of the basal ganglia, not too surprising with exercise. The mitochondria are totally lighting up here. That's kind of cool. Remember, if we, based on everything we know, probably one of the big things that's causing is going to be probably an astrocyte that's happened upstream. It's probably something happening from astrocyte, probably from lactate as a key player. And also the other thing we see that is in the motor area, not too surprising. Again, this is, you know, if you're using the motor systems, you would expect that. There we go. There's the cerebellum. And remember I told you that the back of the brain is getting highly recruited as well in Parkinson's disease. Actually, some of the data we have is that this is more so in Parkinson's than you see in non-Parkinson's, suggesting back to the point that I said, this is a systems problem. I'm using as many systems as I can. And isn't that interesting that we can show this by simply where the fuel is? Isn't that cool? Mm-hmm. So we're being able to prove this by going, look, something's happening here, guys. Energy is being delivered, and these mitochondria are on fire right now, trying to recruit and work hard because they're needed, because they're being told to get activated. And then finally, the other part we have is this. Yeah, there's an area in here. This is the prefrontal cortex. We caught a part of it in the animal. This top part here, red, is executive part of the rodent. So the point is, we're seeing the prefrontal, we're seeing the executive system, motor, we're seeing the striatum basal ganglia, and we're seeing the cerebellum. And this is the difference. No exercise. So that's the other point. This is Cannon's law right here in action. If I am not doing anything, why would the brain know that I need energy? It's really the signaling between the neuron, the astrocyte sensing this, that there's something that this brain is now trying to do that says, I need help here, right? My argument is that, yes, there probably is a brain-body connection to this whole thing. And lactate is just one. People have mentioned other molecules that might also be playing a role here. But lactate allows part of that signaling to come in from the periphery. Also, me using my brain is also creating this effect as well. You understand?
What happened when you injected the mice with lactate?
Yeah. Thank you. So when we inject the mice with lactate, we also see an increase in trophic factors. We do, but we see no evidence of synaptogenesis.
Say that
Well,
again. I'm sorry.
when we inject lactate into the brain of animals, we see an increase in trophic factor expression, particularly in the area that so we see an increase in trophic factor expression. We can see that. And we can see it's still coming from astrocytes, by the way. But we don't see synaptogenesis. We don't see new connections. We don't. We only see that when we exercise. So, but that's such an important point, because I think it goes back to the idea that it really has still to be activation of the neuron. At the end of the day, the neuron is really calling the shots in saying that I need this breath, I need this homeostatic range. I'm driving this, right? And I think also, to your point, it really speaks a lot for these sorts of studies that would be done without that. What would it know to do? If I give trophic factor, if I give, stems, whatever, it doesn't know what to do with that unless I drive the system by Cannon's law. I need homeostasis. I need a range so that I can accomplish what I want to accomplish.
It's sort of like throwing manure into your garden and not having the tomato plant there. So if you just take lactate and sit on your butt, just like taking manure and throwing in your garden, if you don't put seeds down, you're not going to get a tomato plant. You need to have the template. You need to have the downstream target of the benefit of one relation. It's sort of like, going on a date by yourself. It's like, yeah, I can do it, but it's certainly not going to have the recipient who's going to tell me what a wonderful conversation. You still date. So you can see that you need both. You need both happening. It's a balance, a teeter-totter, or like a teeter-totter by yourself. It just doesn't work.
Yeah.
But you put something a target, then you get this relationship. And the relationship between the astrocyte and the neuron can be established only if the astrocytes are activated, lactate, and the neuron is activated, moving and running and thinking.
Yeah.
You can't just lie there.
Yeah. So I'm going to end right there, and then we're going to just take questions. Oh, great. What, yeah.
I want to thank both of you for coming down and doing this presentation. It's really important for us and it's our community here. But I wanted to ask the doctor, when I go on a long walk with my wife, say about 4 miles, at about the 3 mile point, I often turn to her and go, I feel the way I used to. And is it the result of the energy slide that you were sharing with us? That's what's driving that feeling, do you think?
No, it's the wife.
Good.
It won't work with other women. No, I think Giselle and I, and actually Giselle, if we had another 2 hours, we'd talk about it. Not your wife, but we'll talk about the idea that we think of parameters of exercise. And we're doing a lot of thinking of that right now. And Giselle's writing some papers on this. When we think of exercise, Claire comes in and she says, okay, we're going to think of parameters. Okay, duration. Can't I just exercise like can I do a 20-minute exercise turn up the speed so it's 7 minutes? And do I have to exercise? You saw that paper. There's an article in the New York Times last year. You only need to exercise like 3 seconds at a time, right? That's
Like 7
I'm
minutes.
sorry, but
Right.
that's stupid.
But I think it's really sort of slightly different.
No, but duration. We think of duration, you think of intensity, and you think about all these parameters that you do in all your exercise. So when you're going on a 4 mile walk, by the time you hit this avenue of, or this threshold or plateau of intensity, it may take mile 3. So this is why when we are exercising, you need to have a certain degree of duration and intensity, things of that get the bloodstream going, get the lactate being produced, get the activation of those circuits, and then that's the benefit from it. And I think this is something that we try to achieve. You want to talk about bouts or anything, or?
Yeah, I mean, yeah. I mean, no, that's, no.
Yeah, okay. So it's really the intensity.
Yeah.
So it does take time. And then,
Intensity
yeah.
does matter and how long you stay at that level. Exactly, because I think as Mike was showing on that slide, I mean, the idea is that you are getting kind of a transfer, a little bit of energy and, yeah.
Well, it's all my doing. It's my wife's doing, so.
Yeah.
But we all feel better when we exercise, no matter what state we're in. I mean, I feel better when I exercise.
Yeah.
Next day after a run, I feel much better.
Yeah.
So I think.
How long does the benefit last?
How long does it benefit?
How long do you get the benefit of eating a burrito? How long do you get the benefit of that one burrito?
Okay.
Oh, so what do? Do you go to get another burrito?
Yes.
Another burrito? Another burrito for the rest of your life?
You gotta keep
So this is something that we've argued with reviewers or our grants. They're like, exactly that question. How long does the benefit last? And therefore I can stop, right? No, the benefits of eating stop at so many hours and you got to eat again.
Yeah.
So exercise becomes a permanent part of our lifestyle. That's really what
Yeah,
we're looking
that
at.
is true, but
I guess the question is it once a day exercise or twice a day?
got it. So we'll get to, I'm going to tell you that in 1 second, just bear with me. So one other point though, I think in terms of, so we all agree that we have to keep going, right? That this is a commitment and that the brain is really, we would argue is built for this, right? We want to maintain that homeostatic range. That means you need to keep challenging it. We get that. I think the second point though is stopping. There are a few studies that have done this, which is if we're interested, for example, looking at trophic factor levels as a output of something. Let's say we think trophic factor is a good marker that you're working hard and we can see trophic factor levels increase. What a group actually here in Irvine, Carl Cotman did was he put some animals aside. They had been exercising, saw trophic factors climb, and then put them aside and waited a couple of weeks. And he had that same group that had rested, he had them start exercise again and compared to a group that had never exercised. And what he showed was that the trophic factor increase was faster in those animals that had been exercising. So there is this idea of priming, the idea that having exercise gives you, it's not like it's completely lost. It does prime the system for the next time, sort of like a memory of something. I think in terms of frequency and duration, it really depends on, I think, what aspect of exercise you're trying to get the benefits from. And I know that's kind of not what you want to hear, but I think from a metabolic point of view, we think about, how much kind of energy support we want in the brain. This whole idea of 150 minutes of intensity. I mean, you guys have heard that 150 minutes of moderate to vigorous intensity
per week.
Per week, yes, per week, thank you.
That's it for
Day, that we think that's on the low side.
Yeah, that is on the low side. 150 minutes of moderate to vigorous intensity is really talking probably about the energetic aspect of it. If it was just, if you purely took the idea of energetic support, that would be, and the idea there with 150 minutes is doing it any way you want to do it. You could do it, one big bout, or you could do it in little bouts throughout the week. But it's just this whole idea of every week I get about 150 minutes of at least moderate to vigorous. People ask, what does that mean? That means you're breaking a little bit of sweat. You may not be able to talk as fast as I'm talking right now. So you might have to take a breath, that sort of thing. So that's from the, I would say, metabolic idea of what exercise may be doing. But from a skill level, where we're really trying to activate certain areas of the brain, particularly things like cognition, then that's going to require some element of repetition. And in those sorts of studies, at least, that's been at least 3 times a week minimally, where I'm getting practice and repetition, because that's what skill and cognitive engagement requires. And probably, again, because usually skill practice, you're also trying to learn this. That means you probably want some break in between, so that you're getting some sort of retention and then building on that. So I think most of the studies in terms of skill practice, which we also are big believers in, because we believe that's really directing where these benefits are happening, like cognitive strategies, at least 3 times a week. Claire, you're going to say something.
Oh, I was just going to say, I forgot actually what I was going to say before, but, what we tell people is a minimum of 3 times a week is really what the research has shown to be beneficial, but that likely each additional day isn't beneficial, that exercising 7 days a week is a great idea. It shouldn't necessarily be max intensity every day, and maybe it should be different things on different days to keep it interesting for your brain and for your body, so you're not just using the same muscles every single day, but that, somewhere between 3 and 7 days is, the goal. And then it kind of depends on the person and just their real life and what fits into.
Yeah, I mean, how many people here, just from that point of view, how many people here have read Malcolm Gladwell's Tipping Point, right? And so the idea here is practice is everything. And you can't lose for more practice, right? Unless you're doing literally the same thing over and over again, and then you're not learning anything. So I think when we talk about cognitive strategies, right, and learning, the more practice, the better, right? And so, as Claire said, 3 times a week is probably the minimal amount of repetition and practice you want. But, the Beatles got good because they weren't practicing 3 times a week. They got good because they were practicing every day. So as Claire mentioned, more is better. And when it comes to things like cognitive strategies and skills, it's everything we know about learning. It's practice, it's challenge, but more importantly, learning is about problem solving. If you keep remembering that, am I problem solving? Then you're learning. If I'm doing the same type of practice with exercise over and over again, and I'm not problem solving, moving it to the next level, then I'm not learning. So that whole idea of what learning is about problem solving, right? Challenge or intensity, really all that boils down to is problem solving. More practice, you remember, oh yeah, the Beatles every day, right? Yeah, the more you do, the better you get. I think that's the bottom line. And again, that's kind of mixed in with this whole idea of kind of what we would say just, working out hard, this intensity level, which has gotten a little bit confusing because people say this is all aerobic fitness. We all hear that, right? It's all about, we didn't bring that up at all, right? This is not about oxygen. This is about fuel. And the idea that lactate, the harder you work, you're breaking down different types of fuel. And obviously lactate is going to start being play a bigger role in that, the harder you work out. Is sort of what we're talking about here too. But the truth is, I think you're also realizing it's not just one thing, right? I mean, I think all these things are playing, some role in these benefits to the brain. We're trying to also emphasize it's not quite that simple. Again, what Mike said, it's not a glowing brain. I mean, our brains don't glow after. It'd be nice, but it doesn't work like that. And we do have some very finesse and specificity of these effects. And we like to think, at least everything we understand, is because we're using our neurons in a very specific way. There's a reason why the prefrontal cortex lights up. I'm using cognitive strategy, right? It's not that other areas aren't, but that's probably a big one, right? Just like I'm using the basal ganglia as I'm activating or getting busy. And now, as I said, we kind of suspected the cerebellum plays a role, and it seems to be the case. So that's creating the specificity of that effect. the more I do it, the more I'm practicing, we would imagine that these neurons are firing now, firing, demanding more support, looking for that fuel, right? That energy support. So it's that marriage of the practice and then turning on pathways within themselves and to be the recipient of lactate. Now, they all get some, but the idea is that these transporters start going up or the receptors start climbing that are providing a bigger signal to say, come here and work on this side, if that makes sense. I keep doing that every day. It's a concert that's happening harder and harder. And as Mike says, it's also building in some sort of long-term relationship so that these things aren't just a pass in the night, but they're actually now secure and creating some type of connection that's building in resilience and increasing the range of my homeostatic state. Does that make sense? Okay. Yeah.
I'm wondering where fatigue and recovery would play into this, because I work mainly in personal training and physical therapy as well, and a lot of the time you see that yes, doing more is better, and there's kind of a scale where you'll come up before you'll plateau, and then you change duration and intensity. But if we're talking about the brain as like a brain injury, like for Parkinson's, and believing of that system that there's a recovery that we're going through as we're working our way back up into exercise and retraining the brain in different ways. How do we tell from studies like this where it's appropriate to rest?
Yeah.
And let the brain recover?
Right.
Is fatigue an example of like our body telling our brain, that brain body connection of like, I'm not providing you energy?
Yeah, totally. I mean, I'm sure everyone would have a different point of view. I'll say a couple things. I mean, in my mind, I think the first thing that we think about is dopamine levels being low, and that they're due for dopamine, right? And there has been a little work that's been done, again, mostly in animal studies that have looked at the question of, learning and practice in the context of very low dopamine levels. And the idea is that that's probably not optimal. You don't get the functional component of the synapse is not as healthy as you like it to be. So I think there's that component, right? So you need to make sure that certainly dopamine levels are adequate, and that may be part of when one should be practicing, as an example. I think the other thing that I've often wondered about is, I think, and obviously muscle fatigue and all that, too much lactate building up, you're not able to clear it, all that sort of stuff, giving a chance for it to clear. I think the other thing, though, I from a systems point of view, I've often thought that, because I definitely have patients who prescribe to me fatigue, and fatigue is a very difficult thing for us to treat, by the way. People get, and it's a big symptom of Parkinson's. I've often wondered whether fatigue was to some degree overuse or extended use of cognitive strategy. I mean, if you think about it, using cognitive strategy a lot, is tiring. Having to plan out everything and think about everything you're doing is exhausted. And I've often wondered, only because I felt it myself, when I would go on a treadmill, or something, and I would have to be thinking a lot about where, so I don't fly off the treadmill, that I'm on the treadmill, I'm trying to pay attention to everything, I'm fixing my iPhone and all that sort of stuff. Oh my God, after 5 minutes, I want to get up, I'm so tired, and then I kind of like get through it, I feel better again, right? And I've often thought, it's probably because I'm overthinking this. I'm totally overthinking this. If I could just like zone out and, stare at that and just not overthink this yet and just kind of let my legs move, I feel like, oh, I can power through that faster. So I definitely think the whole idea of using cognitive strategies a lot is exhausting. And that's just one thought I had. Claire, did you have any other thoughts?
Yeah, I would agree with that. I mean, I think the medication optimization piece is huge. And we talk a lot with people about that, because a lot of people who do want to focus on exercise as a treatment, in some cases want to really try to minimize medication. And I understand that, but just like not fueling enough with food. I mean, if you don't eat enough, you're not going to be able to exercise well. And if you don't have enough dopamine, you're not going to be able to exercise as well either. So I definitely think that's a huge component. And then otherwise, I definitely think like the more that people have to be thinking about the exercise they're doing, that can be tiring, and physically it can be tiring. And so it's just depending on the person. Sometimes people need different forms of exercise, some that are more or less cognitively demanding, and some that are more physically demanding, but kind of simpler cognitively, like a bike or something like that. or really get the blood flowing. But my experience over 16 years is most people, like if you can get them over a threshold in terms of the amount that they do per week, fatigue starts getting a lot better. But so many people are just below that threshold. They think they're exercising, doing 20 minutes a day or doing a few times a week, and it is just not enough to get over that threshold, the tipping point, to really experience the full benefits of exercise.
Mike, you want to take one?
No, let's hear it first.
Okay, how about over here?
Have you had anything in the relationship to diet and production of lactate? And can you change your diet and increase your life?
Yeah, we have a multi-million-dollar grant that went in, that's on addressing diet in the brain.
Yeah.
And we do know it's extremely important. There's
Yeah.
no question. And so we'd like to try to better understand the relationship between diet and optimal brain
Yeah,
health.
but not by the lactate per se. I mean, there's other nutrients that are really good for mitochondrial effects. So they're working in concert with lactate and astrocyte function that are being supported by micronutrients. Some of them are antioxidants, a lot of antioxidants. One big role that astrocytes play on mitochondrial function in Parkinson's is that they supply a number of antioxidants to the brain. And what that means is in neurons, what we typically see as part of metabolism is a buildup of oxidative stress. What they are is these are oxygen molecules. They're part of the normal cell activity in metabolism. And they have these oxygen molecules are highly reactive. They have extra electrons that allow them, to be very reactive, and they can tear up the cell. And for all of us, for most of us, we have some capacity. Again, this is the homeostatic range to manage them. There's a lot of pathways and a lot of other molecules that are present that clean them up and get rid of them. In Parkinson's, it's thought that they are not being as well regulated. And astrocytes may be one of the roles for that, because astrocytes provide, again, so many things they do. One of the things they provide are the antioxidants that get rid of it. So we think that diet offers one way to get more antioxidants into the brain. Obviously, we hear a lot about vitamin C. Those are natural antioxidants. The diet field so far has, I think, convincingly demonstrated that really the best way to get in the sorts of things that would be helpful for mitochondrial function and antioxidants are really through diet. That really supplements by themselves don't do enough. That things do better. Kind of the message today is always about context, right? So people, at least the diet field, seems to really demonstrate clearly that if you're going to focus on things that are good regarding diet, you should eat, as opposed to supplements, that sort of thing. There has been several studies that have gone on. A big study that came out of Rush and Harvard looking at the Mediterranean diet. We all hear about the Mediterranean diet. But more recently, and the Mediterranean diet was developed more for heart health. But in terms of brain health, there was a lot of interest in really focusing on diets that had anti-inflammatory context as the Mediterranean, but antioxidants for the reasons I've just mentioned. And so they came up with a diet that's called the MIND diet, M-I-N-D. It's a derivative of the Mediterranean and the DASH diet, and it really focuses on antioxidants as well as anti-inflammatories. The other nice thing I really like about it is there's a 15 items, 12 or 15 items that are clearly laid out. There's no gimmick to this diet. You just kind of follow the general recommendation of the diet. You print it up, you slap it on your fridge, and you just kind of pay attention to what you're eating during the week. Number 1 and 2 on that diet list is green leafy every day. Blueberries, very high in blueberries, cutting back on saturated fats. Grains, lots of grains, as much as you can through the day, at least 2 or 3 times a day, grains. Obviously, sugars are simple sugars, not all sugars. Complex sugars aren't bad for us. Sugars we get in through fruit and stuff is not a problem. It's when you're literally pouring sugar into your body, right? That's not good for us. I don't know, right? Somehow we thought that we were okay. It's not. Don't do that. So that's going to be soda. Yeah, don't even think about it. Just don't even think about it. Processed food is just bad. And anytime you go out there, we always laugh when you see the McDonald's commercials saying, and here we have your healthy salad. I don't think so. Okay. Processed, there is no, it's an oxymoron to say healthy fast food, right? There is no healthy fast food. So we say, just don't do it. No fast food. No, nothing regarding to diet, any type of, and the diet, all the sugary substitutes are bad for us too. So just don't do it. right? So, I mean, I think it's everything,
Sweeteners, yeah, artificial sweeteners to stay away from.
artificial sweeteners.
I mean, I used to think that this is all just like, oh, BS. I mean, they just want to say that because it's not natural. And then I spent, a couple of years really reading and understanding the literature. I had some colleagues that study this, and it's not good for you. It's really actually detrimental. And in fact, there's a very close relationship between the brain and eating that is really kind of weird, actually. And one of the things that I find that's really important with sweeteners is that they fool your brain. And the really easiest example, and I never thought of this until someone said it to me, and I said, wow, that's really interesting. Anybody run marathons? Excellent, got somebody. Do you ever get thirsty?
Mm-hmm.
And when you get thirsty, that you get a little water,
Mm-hmm.
right? And you drink the water. And as soon as you drink that water, how long does it take between drinking the water and going, oh, I feel so much better?
During a marathon? I don't think I've felt that.
Okay, you never feel good during the marathon. No one ever does.
But right after the
But
marathon.
right at the marathon,
Uh-huh.
within how fast drinking water do you
get
feel
relief.
relief?
I don't know.
Soon.
How fast?
Quick.
An hour?
I don't remember.
Okay.
It's like
It's
that,
been a few
right?
years.
As soon as Johnny, you drink water, you
Seconds.
feel,
Instantly.
you feel pretty good, like, wow, I
Instantly.
feel better.
But if I were to look at your brain and all that machinery inside of you and say, when is there a physiological benefit to you drinking that water? It's like 8 minutes. And you're like, wait, but I feel good right now, right?
Yeah. So there's a
What's happening
delay.
is that the brain is actually overtaking how you physiologically feel to what you know is going to happen. So when you take a sweetener, and I never thought of this until recently, when you take a sweetener and you drink something sweet like a Diet Coke, the brain says, thank you very much. I'm looking forward to the rest of that mango, all that complex carbohydrates,
That are
and all
good
those
for
things
you.
that are going to feed me.
Yeah.
I really feel happy. And then what happens inside the body? You don't get it. It's not there. You have screwed around with Mother Nature. And that's actually not good. And we now have evidence of changes in the brain that occur in the context
It's
of
like a
that.
tease.
Yeah.
It's like a tease. It's never coming because
Don't tease Mother Nature.
there's no
Yeah.
real nutrients that are going to follow it. You follow me? So the brain is getting prepared for this great, very healthy micronutrient experience that it actually never gets. Kind of mean, Don't do that.
Yeah. So
Yeah, the question in the back, yeah.
Dr. Petzinger, we came to see you about 10 years ago at SC. This is my pain, my husband, and he has had Parkinson's for 50 years now. So my question is about NOH. It didn't really come into play until this last year.
What did?
Orthostatic.
Oh, hypotension, yeah, orthostatic hypotension.
There's been a break for Mike with exercise, with different things that he's gone through.
Right.
We've been with Claire since she started.
Right,
So my question is, you're talking about stabilization
right.
and stabilization of the autonomic system and stabilization of blood pressure. And we have gone to so many others, maybe it's come back to you, but the thing is not many people could help me. And
Right.
so he's on droxidopa now.
Right, that's right.
But, and how often I had to ask somebody what the half-life was.
Right.
That, they're telling me every 6 hours,
Right.
he's fallen off and fainted.
That's right.
So I'm wondering, if you've ever seen, I'm sure the more you exercise.
Yes.
The better the stabilization.
Yeah, that one's a trickier one. That definitely is trickier because the autonomic nervous system that gets affected in Parkinson's, it's probably central and peripheral. The autonomic nervous system has 2 components to it. The dorsal motor vagus, anyway, the dorsal motor nucleus of the vagus, the vagal nerve is one of the examples of what's affected early. So even though you may not be having a lot of problems with it, it's actually a very early problem in Parkinson's. This is what constipation is about. We don't know how to fix that. I mean, we treat it symptomatically. It's very challenging, and there's very little known about how, if anything, what it does to that system.
Intake of like the sea salts, making, boosting.
That's right, and the droxidopa is working centrally, and then we use fluid, we use long socks, we use compression socks. Sometimes we have to, and then I mean, droxidopa is relatively newer, last 9 years, and florinephrine and midodrine. Yes. But over time, everybody gets that problem, some a little bit more than others. But the effects that exercise have on that, not clear that it does anything good or bad, other than that the person is always having that. I think the other thing we pay attention to are the medications, because the medications can certainly make it worse. We think about the dopamine replacement, certainly the anticholinergics, amantadine, can make it worse. So we try to minimize all that, keep
You
things.
just off all of that.
Yeah, right, exactly, because they can make it worse, but it's supportive there's no question. I think the autonomic nervous system and the autonomic problems that we see is definitely one of the hardest for us in terms of managing it, because there is nothing that really, it's supportive care.
Well, I mean, and I noticed too with, lots of high reach, I'm not diet.
Yeah,
It's gotta be.
yeah.
And not a.
Yeah. Yeah. But I think also, and obviously playing a role in the bowel. I mean, we think that that's one of the contributors probably of things like weight loss. So we see constipation on one extreme, that's the autonomic nervous system. We can see weight loss as well because of probably about poor absorption. Just the gut motility is not great. So you start getting poor absorption. Obviously, we use diet to try to manage that as much as possible, but it's not perfect. I think it also starts impacting, without a doubt, how quickly these drugs work, right? Because if you have poor absorption because of poor motility, it's going to take forever for drugs to take effect. And we see that in that we see, people have shown a buildup of the pills in the gut. Hopefully, in terms of that, I mean, there's 2 pumps that we're waiting to see if they can, when they're coming out. The pumps you might have heard about is actually a form of subcutaneous carbidopa levodopa that's being administered subcutaneously. I've got a couple patients on them. And so you're usually filling up the pump for the day. One of the challenges and why it's taking so long to come to market is that L-Dopa just doesn't like to be in any form other than oral. So, when it goes through the skin, what they're finding is some scarring that's happening with the pump, and that's what is slowing down it going to market. Because if you have a scar, then you have to find a new place for the pump. So they need these scars to go away faster. So they're trying to figure out how, probably from an engineering point of view, how to get the drug a little bit more dispersed, so it's not coming in through 1 port or even 2 ports. So disperse it a little bit better, see if the skin tolerates that a little bit better, and or some other type of chemical that might break down the scar.
Are you experiencing that skin to be thicker and tougher?
Yeah, it just isn't work. Yeah, you can't put the pump there again. Yeah, so that's a challenge.
I was just gonna say, for orthostatic hypotension, we definitely see a difference with exercise.
Okay,
So I
that's
think
great.
aerobic exercise, it has to kind of be modified for the person depending on the day and how they feel. But we'll take people, we have people coming in who are like, oh, I feel a little lightheaded and their blood pressure is like 75/40. And at most places, like at cardiac rehab, you get turned away with a blood
Right,
pressure like
right.
that. We do a bolus of 17 ounces of water and then usually start with a bike so that it's safe and people aren't going to actually pass out or anything.
Yeah,
But we definitely see an improvement at that time with exercise, when we take it later between
Yeah.
the hydration and exercise. And over time, I definitely see a difference. It's a study I would love to do
Yeah,
because
that's
I feel like
great.
it's such a big
No,
challenge.
that is.
And I feel like it makes
is.
sense that exercise would be beneficial. It's helpful for the cardiovascular system and the neurologic system.
Yeah.
I have to say it clear, right, because I see the difference in my home, that he's more stable.
Yeah,
And
that's great.
his blood pressure is stable from like noon, 1 o'clock, and when I take it when he's laying down at night, and because it's a black
Because,
box warning,
right.
but he hasn't gotten to the point.
Hypertension or anything like that, yeah. So it's more stabilized, yeah. Yeah, no, that's great, yeah. But to Claire's point, there's much less data on that, for sure. And, but that's great, that we should do that.
Yeah, I would like to.
Okay, yes.
I'm just trying to formulate this question with limited knowledge.
Yeah, that's,
So what I saw that was important were the neurons,
yes.
and that with Parkinson's, are the neurons dying?
In a certain part of the brain it is, but not in a certain part of the brain, there are some neuronal loss, and that's in the substantia nigra, the dopamine producing parts of the cell of the brain. So that's in the brainstem. In the rest of the brain, there isn't neuronal loss, not neuronal loss, but there are changes in connections.
So is that
So the
where?
neurons are there, but the connections are frayed, and that's particularly true, for example, the basal ganglia. That is true. They're not lost, but the connections are not as robust. Does that make sense?
Yes, is that what neuroplasticity? helps?
Yes, that's right. It's trying to take and make these connections stronger or make more of them. And that's what we call synaptogenesis. And some of the data we showed was that we can see evidence of synaptogenesis. So it's that whole idea of creating more resilience by creating more connections. And again, including creating other areas of the brain stronger. Yes, so synaptogenesis is exactly what we're talking about. Connections, and that would be through neurons. Neurons or what encode information. We also then talked about what the second player would be. What would be a key player? That's the astrocytes. Because the astrocytes are really the ones that are providing the fuel for the neurons to be able to keep doing this. That has to come from somewhere, and that's coming from astrocytes. Astrocytes provide fuel, and they also sense what area of the brains are activated. So this fuel is specific to certain areas of the brain that are where neurons are being active. And they can create a long-term relationship with these synapses. In other words, try to really build and hold on to them by trophic factor support. So they deliver trophic factor and mitochondrial support, things that try to hold on to that connection so that they remain resilient. Does that answer your question?
Yes, and does that mean also that there are new neural pathways?
Right. So new neural pathways, I don't know that we would say new neural pathways per se. These systems are already there. So they're strengthening pathways that are there already, as opposed to creating pathways that don't exist. That they wouldn't do, right? And it's interesting that you bring this up, because I think, even when we think about some of the stem cell ideas or, dopamine cell ideas, what they're asking those cells to do is actually make connections, not even small ones, I mean, long ones. I mean, it is a little bit, science fiction. I hate to be cruel, but I don't see how it would happen, frankly. From everything that we understand about how the brain works, it's like, good luck on that one. Because that's going to, that's a hell of a lot of hope of how that would happen. You see what I'm saying? It's not easy. It's like, so let's see here. This neuron is being, and this whole idea also that I have trouble with is that it's just more dopamine. No, it's not. It's a systems. I mean, dopamine is certainly part of it, but cerebellum has a little bit of dopamine, but it's not that either. Do you see what I'm saying? That's why this is more complex. It's not to say that it's not, it might not be part of something, but the idea that it's that. You follow me?
That stem cells would be the complete solution.
Oh, yeah. I mean, again, from everything we know how the brain works, it's like, really, good luck on that.
Maybe turn that into neurological pathways that kind of create.
Yeah, I mean, just the idea, right? I mean, everything we're talking about is just, this is just trying to get more connections that are already there or make them stronger. We're not even starting with a new cell and like, okay, here's a new cell, I'm depositing it there. I'm not creating any external challenge now. That cell is expected to do what exactly? Branch, connect to another part, hold on. And you're thinking, wow, that's wild.
Yeah, I mean, our work has shown decades ago even is that circuits that were minor in most of your life can become major upon challenge or compensating. So when you lose the dopaminergic neuron from the substantia nigra pars compacta into the basal ganglia, in fact, we showed that the cortical striatal, the front to the as opposed to the bottom, it's up, actually picks up the slack. So you're losing one circuit, another circuit is picking up the slack, and this has been shown in humans and in animal models. So those circuits have always been there, but always been a minor. It's sort of like when a quarterback. So you got what, 4 quarterbacks that dress up for a game, right? And then one plays, but he's been doing that for like all year, and then he gets hurt, and you bring in the minor guy, and he picks up the slack and you win. So the end result is that you can in fact utilize. So Giselle's stressing that's what we need to target.
I think-
Claire's work shows
Yeah.
that's what we need to target, and that's what we
Yeah,
use.
and exercise is really focused on that. I mean, exercise is Cannon's law. It's trying to take what is present and make that working more for us. And again, it doesn't know that it needs to do that unless you tell it, I need it. So make it happen for me. Does that make sense? Nothing happens without a purpose here. The brain simply doesn't know that. It's not going to guess that unless I'm telling it, no, I really do need that. And that's what resilience is. It's really creating that homeostatic range and maintaining it because I'm demanding that you do it. Now, there may be a number of things that we can do to support that. And maybe that's where the cells come in to support a system that's already there trying to work hard, if that makes sense. And I think that's sort of the general feel that we've always thought makes sense. You know, and you know, again, prove me wrong. I have no problem being proved wrong. You know, if you get a dopamine stem cell working and you could make that work, great. You know, that's awesome. But I don't know that realistically, from everything we know, from how the brain works, that it even makes sense. You know what I mean? You can't sit in that. I can't sit in it. I can't sit in a talk. It's actually, I cringe. It's bad. I literally cringe. I'm going, really? How is this supposed to happen? Actually, I was just at a talk the other day, and actually it was a stem cell talk. I know Irvine is doing some work now, but you know, one of the points that was brought up was like, okay, but DBS, I mean, DBS is pretty effective. It's doing, you know, it helps. It's not perfect, but it helps. And they kept saying, but so how is that? It's almost like it's rivaling, it's being compared to DBS, actually. Because the idea is that it's still more about motor. And we've been talking about all these other issues, which it's not trying to address that. It's just trying to address motor. I think the other thing is how long lasting these cells are going to last, side effects for tumors, you know, or dyskinesia. I mean, in other words, it's like, okay, but your competitor is DBS because I can get a good on state with DBS now. And I could turn it off. You know, I mean, it was interesting. I never thought I would hear this competition with DBS, but that's where we're at now. You talk to anybody who's trying to do stem cell work right now in the basal ganglia, they're going, you know, because it's dopamine, right? That's really what they're focused on. And they're going, so why aren't you just doing DBS and what convinced me that this is worth it because of all the possible problems with stem cells, including tumors and all that other stuff. I don't get it. You see what I'm saying? It's so interesting. Again, I mean, I definitely think, you know, that in light of everything we know, I mean, anything that could help support this, I think is good. And I think we do have a lot of tools that we could use to support this resilience, for sure. You know, I just think that we need to be thinking broader. And unfortunately, I think in the field, you see a lot of these really weird gaps, like I call them valleys of death, where people are very isolated in a very singular idea of what they need to do. They almost get caught up on the technology more than the real bigger problem. And yeah, I don't know what the answer is, but I do think, I mean, we like to talk about it like this because we hope, we do. We hear people that are more interested in the systems idea, resilience, and what we would call lower hanging fruit, and more doable, if that makes sense. Yeah.
Exercise is available now, and
it's an external challenge. It fits Cannon's law.
And it's probably the most important thing. And all these other treatments that are being discovered could be
Supportive.
Supplemental and supportive
That's
to the
right.
exercise as opposed to the other way around.
Right. Exactly.
But it takes the exercise to make.
Right, because that's, as I said, Cannon's law.
Yeah.
You need to create the need for homeostasis before you support it. Otherwise, as Mike says, you know, you're throwing fertilizer and you have no rose bush. You know what I mean?
That's for oxygen therapy, and they, because he's a first responder, they gave him 20. So Claire knows this, and so we did 20, and it's once a week, and it's going from here to there, and 8 hours. I don't see a difference. Yeah, I'm not seeing it because we skip the exercise and go do that.
Right.
Time to do.
Yeah.
I just.
That was my
She didn't say
question.
we
What
are
about
not going to get exercise.
Right. Yeah, and it's
Very
putting, yeah,
often.
right. You don't want to put the cart ahead of the horse. You got to.
Is this hypoxic therapy?
No,
I mean, there's both. There's hypoxic therapy that people are using, even to the point where they put this thing on you and they inflate it and it blocks your blood vessels, and then then they relieve it, and you get this, whoa, rush.
No, she's saying.
Right, but so the idea of with either is, can you create a situation that's physiologically reflective of, yes, I'm getting less oxygen, or I'm exercising, breathing in more oxygen. So it's almost like artificially going to VO2 max or VO2 minimum. And you're like, well, that's sort of like putting gas in your car without turning the ignition on.
Well, yeah, that's what she's saying.
And exactly, you're absolutely right, but that they're trying to do both ways, both ends of the spectrum.
Well, but they're actually looking at both,
Yeah.
hypoxia as well, but yeah.
But like hyperbaric oxygen, like if you exercise and get increased oxygenation to your body while exercising and having all the other beneficial things
Yeah,
that happen,
that's
that's likely
happening,
better
yeah.
than a passive
Right, totally.
treatment.
Yeah, I
right?
mean, I think.
Yeah, but we got this amazing tool,
Yeah,
it's called breathing.
yeah, that's true.
And actually, it's a really
Yeah.
fun book that came out about 2 years ago. She wrote this book. She's a exercise physiologist, neuroscientist. I forget where she's at. She wrote a book, and she's like, and she had an article in the New York Times. Here's the app of the week. App. When should I drink water? Oh, it's on my app. All this. And she says, you're built in with the best app in the world. When should you drink? When you're thirsty. Oh my God, I wish I could put that in an app. You don't need it, it's in here already. So we have that. Yes, there are blood monitors, there are these things that give us maybe more precision and stepping and stuff like that. Those are accessory tools that I think can be beneficial, but we are, Mother Nature for the last millions of years has found some pretty good solutions, and maybe we should remember them, and that's what we try to do in our work.
Yeah. Okay, well, thanks, guys.
I love that.